informational webinar. I'm happy to see some familiar faces and some new ones. I would like to give people another 2 minutes to join, so please hold on, and we'll start in 2 minutes. Thank you. Let me see some here. Mhm. Okay, hello one more time, and I think we should kick it away. Welcome to the informational webinar about our newest call for proposals, which is in vitro disease modeling for type 1 diabetes to enhance translational research. And I want to take it away for a quick introduction of Breakthrough T1D for those who are not yet familiar with it. And also for those who don't know me yet, my name is Asi Guzman, and I'm leading the cell therapy portfolio here at the research department of Breakthrough T1D. So let's get started. Most of you are probably familiar with how Breakthrough T1D started. It was founded by two mothers of T1D children in 1970, and what started as a mom initiative has grown so much over the last 50 years, and right now we have a organization that is funding over 450 research projects, and of them, over 70 active clinical trials all over the globe, both from academic and industry partners. And Breakthrough T1D supports the development of therapies in a very multifaceted way. So not only are we supporting research and product development, we're also supporting the regulatory and access aspects of new therapies coming to the field. And by tackling the problems from all the angles, we're hoping to accelerate what is our vision, namely a world without T1D as a disease. So just to quickly go over the interventions that are currently available and being developed in the type 1 field by stages of progression. Of course, Breakthrough is strongly supporting early detection, which is the detection of pre-symptomatic stage 1, what is defined as stage 1 and stage 2 of type 1 diabetes. In terms of real interventions, we have disease-modifying therapies, which are aimed at stage 1 and 2 and 3, and are aiming to delay or restore insulin independence by protecting or replacing functional beta cells. And we have cell therapy for stage 4, so for people who develop dependence on exogenous insulin supplementation, as well as treatments for complications and technologies to support everyday life. For today's RFA, I think the disease-modifying therapies and cell therapies are of the biggest relevance, and I want to quickly touch upon of what these are. So disease-modifying therapies really focus on restoring immune tolerance and preserve the beta cell mass and function through various approaches, including disabling autoreactive immune responses or enhancing regulatory mechanisms, as well as regenerating and protecting beta cells, ultimately combining immune and beta cell targeted therapies. And as you can see here, there are various approaches that are being developed, also trying to reduce beta cell stress, aiming at beta cell regeneration, etc. Cell replacement therapies, on the other hand, are aiming to restore insulin independence by putting back into the body insulin-producing beta cells. And right now, we already have an approved cell replacement therapy, that's Lantidra, and it involves taking donor-derived islets and putting them into the hepatic portal vein and keeping them alive by putting the recipient on continuous systemic immunosuppression so that the graft is not rejected over the course of life. This is something that is being developed further because our goal is to have not a donor-derived cell source, but actually a scalable donor-independent cell source, meaning cells that are being manufactured. And of course, everybody tries to remove the need for systemic immunosuppression because everyone is probably aware what a burden it puts on the graft recipient, making them susceptible for infections, putting a burden on kidney and other organ health. So with that, I think I hopefully have shown you that there is a lot of effort going into the therapeutic innovation, both in DMTs and cell therapies, with immune engineering approaches, with targeted immunotherapies, with development of stem cell-derived islets and gene-edited cell products. But with all this innovation come also translational bottlenecks, and we see a lot of relatively slow clinical progression of the next generation DMT candidates to the clinic. There is a bottleneck in incomplete delivery system, but also lack of early efficacy biomarkers. And of course, it is all aggravated by patient heterogeneity, with some people being responders and some not. And on the cell therapy side, we have the problem of vascularization and engraftment that yet has to be fully solved. There is a foreign body response to islets delivered in extrahepatic transplantation sites. The immune protection sometimes is incomplete, and of course, the cell maturity and function in the recipient body is still an ongoing problem that yet has to be solved in a satisfying way. So I think it's easy to see that there is actually some shared unmet needs between those two areas of therapeutic development, which would really hugely benefit from human relevant in vitro systems. We do want more physiologically representative models in the field that could capture the reciprocal interactions between islet cells and other relevant cell types, such as immune cells, vasculature cells, tissue supportive cells. And we want this development to happen to improve the preclinical to clinical translation of both the DMTs and cell therapies. I think the need for models to screen therapeutics for both immune modulation and cell therapies is really high, but in contrast to, let's say, oncology field, this area of development in the autoimmune diseases is relatively nascent compared to other types, to other areas. So I think in general, the field would hugely benefit by advancing non-animal model systems, NAMs, and I'm sure you're all aware that actually there is a strong initiative from the NIH and FDA to support studies that are relying not only on animal models, but also on predictive non-animal model systems. And this call for proposals is fully in line with this initiative. So the call for applications is in vitro disease modeling for type 1 diabetes, and we called it type 1 diabetes on a chip, but we are supporting advancement of cutting edge organoid, organ on a chip, and tissue, ex vivo tissue systems, all with the goal to improve the translation of disease-modifying therapy candidates and cell replacement therapy approaches. And this is a call for proposals for a 1 year and $300,000 over 1 year that is accessible both to academic investigators and industry partners. And as I said, we're inviting organ on a chip, 3D tissue construct, multicellular co-culture systems incorporating human stem cell derived islets, immune and vascular components, and relevant tissue microenvironments for this call. And I will go in more detail for the examples of topics that are pertinent to this RFA. So here is a list of what we think is highly relevant to us. The testing of safety and efficacy of novel pharmacological and gene editing interventions is really front and central and is something that we want to tackle with this call for proposals. We're interested in platforms to evaluate drug delivery, pharmacodynamics, and combination therapies. Also to identify translational biomarkers and surrogate endpoints that could be used in regulatory packages. Modeling immune islet interactions is another topic of interest. And of course, we're interested to evaluate strategies that are targeting beta cell survival, function, and regeneration, be it within DMT strategies or cell therapy strategies or a combination of those. Because actually, as we're moving closer to translation, some of the DMT assets can be applied for cell therapy purposes in combination with cell therapy products. And I want to go in a little more detail over which projects we have in mind. Excuse me. So as I said, the testing of safety and efficacy of novel pharmacological interventions and of gene-edited cells, as well as scaffold and device materials or controlled release drugs, is front and central for this and the area of biggest interest. For the evaluating of the drug delivery and pharmacodynamics, it's also, as I said, we're also interested in combination therapies such as immune modulating drug release or combinations of immune and beta cell targeting agents in order to identify synergistic or adverse interactions. And this could aim in forming rational combination design for therapeutic strategies that are, for example, using DMT assets to improve cell therapy outcomes. Identification of translational biomarkers and surrogate endpoints also is an area of interest. So organ on a chip or other advanced in vitro systems equipped with integrated biosensors that can capture dynamic readouts of clinically relevant either molecular or functional readouts in real time, and inform discovery of biomarkers that would be predictive of clinical efficacy is of huge interest to the field. And in the future, the obtained quantitative data set should strengthen regulatory submissions and by this provide early mechanistic evidence of therapeutic safety and activity that would accelerate and improve the regulatory path. Modeling of immune islet interactions. This is something that has been very successfully done in non-animal models in other disease fields, in particular in the oncology field. So in our opinion, this is something where the autoimmune field, and in particular the type 1D field, could really benefit from better in vitro models of immune islet interactions. And this would involve incorporating human beta cells, stromal components, and immune cells to model the cellular and molecular mechanisms of the autoimmune attack and tolerance with respect also to, let's say, drug candidates that are aiming to improve the resilience of beta cells to, let's say, cytokine stress. Evaluation of strategies targeting of beta cell survival, function, and regeneration. So I think this is self-explanatory, actually. Microengineered beta cell systems that would recreate physiological nutrient and oxygen gradients of particular importance as we are evaluating various gene editing strategies targeted at improving stress resilience and function of manufactured beta cells. And these gene-edited cells need to be tested in environments that are representative of the transplant locus in human body, and also models systems that would model the transplant engraftment. So we're talking about tissue-specific or body site-specific oxygen gradient, nutrient diffusion, vascularization, inflammation, foreign body response are all of interest. But as I said earlier, this RFA is inviting one-year projects. So we're looking for something that is very, very focused and that is actually building on a established in vitro platform that are clinically relevant, reproducible, and mechanistically informative. So this call for proposals does not look for projects that aim to establish and develop a new in vitro system. Rather, we're looking for platforms that are ready to be rigorously validated. Of course, we focus on use of human cellular components to maximize the clinical relevance, and we strongly focus on validation of the predictive power. So not only should the system be established for use with human beta cells, but it should be benchmarked against pharmacological, genetic, or other interventions from clinical studies. And in the perfect world, it would involve a collaboration between tissue engineering group and clinical partners to link the in vitro findings with in vivo biology. And we strongly urge the projects to integrate clinically relevant metrics, which would be beta cell function, engraftment, vascularization, immune protection of the cells. Because in contrast to, so each field has their own questions, which means the platforms for these projects should be addressing specifically the questions that are clinically relevant to the type 1 field. Tips for a successful LOI. Of course, please ensure that your proposal is aligned with our research strategy, both in the DMT area and cell therapy, and the goals of this specific, very focused RFA. For your LOI, please include preliminary data, even if referencing published work, that would support the feasibility both of your platform and approach and the team, especially if this is a collaborative work. There should be a strong rationale for selection of benchmarks for this particular application and the potential of this platform to accelerate translation of preclinical to clinical therapeutic strategies should be laid out clearly. We do support projects that contribute to the following FDA recommended steps, which is the mapping of critical endpoints, the supporting non-animal model development, right? Proposals that refine and validate already existent systems to predict human response, and that establish a clear validation and qualification pathways. The project should focus on a really rigorous benchmarking against clinical data, demonstrating reproducibility, feasibility, but also and especially predictive power, which is really critical. Topics that are out of scope for this RFA. As I said, the use of general in vitro platforms without specific relevance to T1D is a out of scope topic. So let's say there is a great breast cancer organoid platform with vascularization that you think could be made into an islet platform. This would be out of scope. We are hoping to invite projects that already have a type 1 relevant setup. We're excluding projects that address very broad immunology or diabetes mechanisms without connecting them to beta cell function, engraftment, or therapeutic intervention testing, because this whole RFA is meant in a way to validate a platform for in vitro pharmacology and kind of early predictive kind of clinical surrogate time points and data, and to validate its predictive power and potential to be included in regulatory packages and provide early information on the translational potential of therapeutic targets. Projects that focus solely on murine islets or non-human components are excluded, as well as projects that focus exclusively on in silico modeling without integration into an experimental platform. The funding mechanisms for this RFA are strategic research agreements, which are for nonprofit entities such as academic institutions and can involve one or multiple investigators on the project, and the Industry Discovery and Development Partnership. This is our IDDP program targeted at research and development at for-profit entities. If you are considering applying to the IDDP program, please reach out to me prior to submitting an LOI, and also take a close look at our requirements for the IDDP program, because in contrast to the nonprofit applications, the IDDP program involves matching funds. This award is designed to support one-year projects for up to $300,000, including 10% indirect costs for the SRAs, and the level of funding is, of course, dependent on the scope and overall objectives of the proposal. If you think you have a project that is extremely well suited for this RFA, but is exceeding the budget and the timeline, please discuss this with me before you submit the LOI, and we can talk about the feasibility and the strategic fit. This call for proposals is open to investigators, collaborative teams, and companies with demonstrated expertise to carry out the proposed research. Applicants must have an MD, PhD, or equivalent, and have a faculty position at a nonprofit institution if you're applying via the SRA mechanism. This call for proposals does not have any citizenship requirements. So even if you're located outside of USA, please feel free to apply. We do not make a distinction between applications from different areas. And just to give you an idea about the application process and timeline, the application deadline for the LOI submission is January 28th, where you would submit a letter of intent to Breakthrough, and it will be evaluated and reviewed internally for strategic fit, feasibility, and everything, and be either triaged or approved for a full application by February 11th. If you're approved for full application, you will receive an invitation to submit the full application by March 11th, and these applications will be reviewed by external reviewers who will provide critiques, which will go back to the Breakthrough scientific staff to make the final funding decisions by July. So the earliest anticipating start date of these projects would be October 2026. And with that, I actually want to close the presentation. Again, I'm Asia Guzman. I would be your point of contact if you have any questions about the feasibility or strategic fit or anything regarding the scientific component of your project. Madhu Prakash is our program administrator who is the contact for anything about the uploading of LOIs and other administrative questions. And with that, I would like to close and to open it for questions. Okay, any questions? Please go ahead.
Can you hear me?
Could you speak up a little?
Hi.
Hi.
Hi, I'm Humaira Afrin. I work at the Mayo Clinic. We have a diabetic model on organoid in chip, but mostly focuses on the diabetic complications on the kidney. So I was wondering if it goes with the scope of your RFA.
Yeah, I think this would be a better fit for an RFA focusing on really complications. However, if you are interested, you can always send me a quick scope of your project, and I'm happy to circulate it among my colleagues who are in the complications area and get their feedback for you.
Okay. I think my PI, Dr. Naveen Gupta, has already sent the scope, but I don't believe he has. He is still. I don't know if he has received any reply back.
Okay. Yeah. I will. I will take a closer look, but just from what you told me now, I would say it would be a better fit for an RFA focused on complications.
Do you have a different grant proposal or different RFA for complications?
So we have various RFAs covering most of the areas throughout the year, but also you are free to reach out to a scientist from a research department with a proactive application and just ask for their feedback on the strategic fit. And you can always apply also outside of RFAs. The RFAs are a mechanism for kind of something that is focused that is focused on a specific area. But if you have a project that is very well aligned with our research strategy, but is not a good fit for this particular RFA, please reach out to our research team and discuss if this would be a good fit in general.
I get it. Thank you.
This is Eddie James from BRI. I was
Hi.
curious, how many awards do you anticipate making through this mechanism?
How many what?
Awards. How many projects do you plan to support?
I mean, it depends how many amazing applications we will get.
But it is merit-based.
It is merit-based. I cannot, before we see the full scope of applications, we cannot decide on that.
Okay. I mean, I'm pretty excited about our idea, and I think it's responsive. So I guess I'll send you a one pager, and we can talk about it further.
Yes, please do. Any more questions?
You can also post the questions in the Q&A section or
Yes.
the chat.
Okay. I will stay for some time if anyone will come up with a question. If you will have a question after the webinar has wrapped up, please feel free to reach out to Madhu or me, and we'll get back to you. Thank you, everyone. Looking forward to the LOIs. Okay, I have here a question. If the applicant should have a faculty position, or could a postdoc scientist apply? So we have specific mechanisms for postdoctoral awards, but this call for proposals is meant for people who hold a faculty position. So if you do want to apply, please do so via your PI.
You got another question.
Can for validation studies be performed in mouse models? It depends. It depends what question it is and what response you're validating, because we know that mouse models are more predictive for some responses than for some others. For example, if you want to set up an in vitro system for prediction of foreign body response to a islet encapsulating material, and then you want to validate it in a murine model, I would advise against it, because we do know that the mouse models are very poorly predictive for this. If you want to use something that has actually already been validated in other models wildly and just use the mouse, let's say, to validate the dynamic and extent, maybe, but it really depends on the question you are asking. Oh, I see. Just to check, the question is if you can use mouse models to check the functionality of islets. Yes, you can use, yes, this you can do. So if you are simply using the mouse model to address if the islets you are using can restore glucose control, then yes. The next question is, how much translational do you expect the outcome should be dedicated to? I mean, I'm not sure if I'm understanding the question correctly, but what we're aiming to do is to take an existing in vitro model and either optimize or better even validate it against something that is clinically very relevant. Let's say a novel therapeutics that was shown to improve the hypoxia resilience of cells, or a gene editing approach that was shown to improve the functionality or the immune cloaking of the manufactured islets. And take something that is currently or previously already has been tested in the clinic or in late preclinical models such as NHP or pig, and see if your platform is able to reproduce this result. So if we want to validate the accuracy of prediction, the predictive power, and the physiological relevance of existing models, and also identify the necessary optimization steps that would make it potentially even more predictive, so that the readouts can be used for in vitro pharmacology for complementing regulatory packages. I hope that answered the question. Another question is, does it matter if the cells used to be studied do not originate from human tissues, but maybe porcine NHP on mice? We strongly prefer human material if possible. Okay. I think this is probably it for today. Oh, sorry. Is the structure milestone driven is another question. No, the structure is not milestone driven, but because of how short the timeline is for this project and because of how focused we want it to be, we suggest something like having 1 or maximum 2 killer experiments over the course of the year that would really demonstrate if the platform is or is not predictive of whatever you are trying to model. Okay, I think we are done with the questions probably. And if you will have any after this is wrapped up, as I said, please reach out to Madhu and me, and we'll be happy to help you. Thank you so much for attending, and I'm looking forward to the LOIs. Thank you. Have a good day. Bye.